Okay, so let's talk about this neural physiology and what's going on in the actual neuron that's allowing us to send this nerve impulse, or what's keeping the nerve impulse from being sent. Okay, so first thing we want to understand is there are three types of potentials that are occurring in the neuron. The one that we probably talk about the most, and that most people are most familiar with, is what's called the action potential. And that is literally the nerve impulse. That's the main purpose of the nerve, to be able to send this signal to basically cause the release of neurotransmitter. So when we were looking at a neuromuscular junction, we saw that the action potential came down the axon first and then went to the synaptic bulb, where it changed the voltage so calcium channels could open up and calcium could come in and trigger the exocytosis of the neurotransmitter. Okay, now that's a whole different subject, but same basic thing is happening here, and actually the same thing is happening here. Okay, so the action potential is going to occur starting at the axon hillock, where the axon attaches to the cell body, and it's going to generate down the axon. Now, this axon is myelinated, so in that case, the true action potential is only happening at the neurofibril nodes, also called the nodes of Ranvier. But if the myelin wasn't there, then the action potential would occur the whole distance and then trigger the synaptic area, the area where it ends. Say this is a continuation of that axon we saw, then it's going to eventually run down here where we're going to release our neurotransmitters to another, in this case, another neuron. So again, even looking at this, you can see all these different axons synapsing with this one neuron. With there's the soma, or the nerve cell body, and look at all these different axons that are synapsing with that one neuron. But anyway, we'll come back to that idea in a few minutes. Okay, so this first potential, action potential, that is actually what is going to be triggered by reaching a voltage called threshold. Now you'll see different voltages used for that number. I usually see either -40 or probably even more often -55 millivolts. So again, once we reach a voltage in this area, then we send the action potential. If we never reach threshold, we never send the action potential. All right, we'll come back to that. The other two types of potentials are, well, what's going on in the cell all the time? In other words, if I measure inside the membrane anywhere in the cell, what would my voltage be? And that's what we're going to call the resting membrane potential. So looking at this chart, even notice I've got this thing here. It says resting potential, and it's around -65 millivolts. So normally we see that -65 to -70 millivolts is the range of a resting membrane potential that's found in a neuron. So anywhere I'm measured in the cell when I'm not going through the other two potentials, then I would have a resting potential, which would be about -65 to -70 millivolts. So even another graph we're going to look at that will be really important for your test to understand is, notice here I have this -65, which is my resting membrane potential. Now, the last potential is what we call the graded or synaptic potential. And the graded or synaptic potential is literally a potential that's occurring where a synapse takes place. So notice I've got an excitatory and inhibitory synapse taking place. One of them looks like it's right where the dendrite and the nerve cell body is. One of them's right on the cell body. So where do we synapse from one neuron to another? Well, we always do it in what we call the receptive areas, which is going to be the dendrites and the nerve cell body. You're not going to see a synapse coming right here to the middle of that axon. The only place in an axon that may have a synapse could potentially be the very end. We could potentially have a synapse going to another bulb area, but you're not going to see it on the rest of the axon. Okay, so I have three potentials I need to understand. And they're these things again, graded potential. It's called graded for a reason we're going to talk about later. But graded potential is basically what's given me the excitatory or inhibitory signal. Okay, and if that excitatory and inhibitory signal, once I take the average of both of those, and I call that integration, if that signal that is occurring because I'm releasing neurotransmitters here, and I'm allowing ions to come in. If I'm allowing positive ions to come in, I'm going to get an excitatory signal. But if I'm allowing positive ions to leave or negative ions to come in, then I'm going to get an inhibitory signal. Now, my resting membrane potential is what I can measure no matter what's happening. But when I start measuring the voltage when I'm opening up doorways with my neurotransmitters, then my voltage will either rise towards threshold or it will fall below threshold and maybe even below my resting potential. Now, as it rises towards threshold, especially after its threshold, we call that depolarization. If my signal causes me to drop below my resting potential, we call that hyperpolarization. Okay, and of course, we're going to talk about repolarization, the other part of the action potential, a little bit later. Okay, now, so again, I have three potentials: resting, graded, also called synaptic potential, and action potential. Okay, where do they occur on the cell? Well, resting occurs everywhere. Graded or synaptic only occurs where the synapses take place, which is in my dendrites and nerve cell body. And action potential only occurs starting at my axon hillock, where the axon attaches to the cell body, throughout the rest of the axon. Now, the next question is, what causes each of these potentials? And there are some videos I'm posting that talk more about the detail of this, but let me just mention them now. Resting potential is caused by what we call sodium and potassium leak channels that are found everywhere. Now, because it is such a negative number that makes up our resting potential, what that means is that there's a lot of something positive leaking out. Well, what is it that is leaking out at all times? Well, what's leaking out at all times is potassium. Well, since potassium is a positive ion and it's leaking out, then it's going to give us a very negative number. Now, so I have a bunch of potassium leak channels all over this cell where potassium is constantly leaking out. I have a few sodium channels, because if it were just potassium channels, this number would actually be around -90 or so. It would actually go down until potassium reached equilibrium on either side of the cell membrane. We'd have as much potassium on the outside as inside. And of course, if we had as much on the outside as inside, then the leaking would stop. All right, but we do have a leakage of potassium, which helps maintain this very low number. But I do have a little bit of leakage of sodium into the cell, which keeps that number from getting even lower. All right, now, in each individual channel, though, let's say I just had one channel that allowed both potassium and sodium to leak, then more sodium would leak in than potassium would leave, and that's because of something called an electrochemical gradient, which I've explained in another video, so make sure you listen to that if you're not sure what I'm talking about. All right, but back to this particular picture. So what I've got here, I've got my resting membrane potential, which is occurring everywhere. But if I want this cell to send its impulse to release its neurotransmitters, I have to generate an action potential. Well, the only way I can generate an action potential is if my voltage inside reaches this magic number called threshold. Okay, threshold is the number at which voltage-gated channels start opening up, and then they produce a positive feedback, because once they open up, they trigger the next group to open up, which triggers the next group to open up, which triggers the next group to open up, because each time they open up, they bring in enough positive ion where the ion flows in this direction, it causes the next area to reach threshold, which then opens up sodium channel. Sodium comes in, causes the next one to reach threshold, and it keeps generating. Okay, now what is it that causes me to reach threshold? Well, what we see here again in this chart here, where I have an excitatory signal or inhibitory signal, that then, the excitatory and the inhibitory signal is going to be what is going to be produced by my graded, also called the synaptic potential. Okay, now again, my resting membrane potential caused by leak channels, my action potential caused by voltage channels, and that's the voltage that right there is specifically the voltage. And again, you may see that as negative 55, but we won't get caught up on the numbers as much as the concept of what the numbers mean. Okay, but once we reach that, then it's like an all or what we call an all or none event. Once we open it up and we hit threshold, then the impulse is sent forward and it doesn't stop anywhere in the middle. It keeps going till it gets to completion, a classic positive feedback loop. All right, but what caused us to reach threshold was our graded potential, and that's caused by chemically gated channels. In other words, the channels that literally have, now look, here is exocytosis of my neurotransmitter. There it is going to my chemically gated channel. There's the chemical going to, I call the chemical the key, going to the keyhole, which then opens up the doorway. Now my ion can flow through. Since sodium is a positive ion, then if I have a bunch of positive ions rush through here, then what that's going to do is that's going to cause my resting potential to go in a positive direction. Okay? And if I get enough positive ions flow through where my excitatory signal and everything else going on allows me to reach threshold, then that will open up my voltage gated channels, and then I will be able to send my action potential. So let's look at that for a second. Okay, notice this graph looks a little bit different from the other because this is actually the signature of an action potential. So notice that I've got my resting membrane potential here, again caused by leak channels, particularly the potassium leak channels. Here I have this area where I went from resting membrane potential and I reached threshold. So this area right here would be caused by my graded potential, also called synaptic potentials, which is caused by chemically gated channels. Now, once I reach threshold, then what I have happening first is depolarization, because now I've opened up my voltage channels. Once I open up my voltage channels, then the sodium starts moving in. Well, since sodium's positive, then notice how this graph climbs. As sodium rushes in, I go from negative 40, and really I started out around negative 65, but the sodium wasn't able to rush in the voltage area till I opened them up by reaching threshold. But once it did start rushing in, notice how this graph climbs. And what's happening is sodium is trying to reach its equilibrium. In other words, the same amount of sodium on one side of the membrane and the other. So as it climbs trying to reach that, then we call that depolarization, because notice I go from a negative number where my polarization is negative. Now as soon as I cross zero, I have depolarized. I've gone from negative to positive. So I am now at an opposite charge as I began. Okay. Well, that can only go for so long, because before I reach equilibrium, once I get up about positive 40, even though equilibrium's more around positive 63, before I can actually reach that, my sodium gates start closing, but my potassium gates, now I've actually reached a threshold for the potassium gates to open up. So now potassium starts leaving the cell, and if we know if a positive ion leaves the cell, then our voltage starts dropping again. And here we see in repolarization, potassium started leaving the cell in those potassium channels, because there are always potassium voltage channels near the sodium voltage channels. They're separate channels, but they're always near each other. So the potassium channels started opening up, potassium started leaving, and now we repolarize. So in other words, we crossed zero again, and we went from positive back to negative. So we depolarized from going to negative to positive. We repolarized by going from positive to negative. What caused us to go from negative to positive was sodium moving in. What causes us to go from positive to negative is a positive ion potassium moving out. Now, even though this doesn't show it, if enough potassium continues to move out where we drop below resting membrane potential, we call that hyperpolarization. And so this graph is extremely important. You're going to definitely need to know it for the test. But more importantly, you need to understand what it's actually showing. And notice that this graph is actually being shown in milliseconds, thousandths of a second. So this is all occurring extremely fast from the point of, our resting potential to our synaptic potential to our full action potential. Notice we're at 5 thousandths of a second.